Hey, what's up, everybody? We are back for lecture number 2 on the immune system. And we spent most of the previous video talking about the innate or non-specific immune system. Today we're doing the adaptive or specific immune system, starting with some fundamental characteristics about it. So the adaptive immune system, it is going to respond in specific ways to specific pathogens. So we're going to see there is not like just a generalized catch-all response, one size fits all, but a specialized response created for special specific pathogens. And this is going to be a systemic response. So the non-specific, the innate immune system, that was very local. Like it would just happen in the affected tissue. But the response to the adaptive immune system is systemic, it is body-wide. And finally, what is perhaps the most important thing is the response improves with subsequent exposures to the same pathogen. So in other words, the response is going to exhibit memory. Okay, in this system, the major cells are the T lymphocytes, which we are familiar with, the B lymphocytes, which we've met as well, and the antigen presenting cells. Now, we haven't used that word like antigen presenting, but we have met antigen presenting cells, as we shall see. Now, let's start with the B and T lymphocytes and where they're produced. Of course, we know they're made in red bone marrow. All blood cells are made in red bone marrow, okay? So hematopoiesis, the production of all blood cells, occurs in red bone marrow. Leukopoiesis, the production of white blood cells, occurs in red bone marrow. Now, coming out of red bone marrow, we will get B lymphocytes that are called naive immunocompetent B lymphocytes. So they're immunocompetent, which means they're able to do their immune system job. The word naive indicates they have yet to be exposed to their specific pathogen. Now, T's are different. T lymphocytes, when they come out of the red bone marrow, they're not full-fledged T lymphocytes yet. These pre-T lymphocytes travel to the thymus first, and it's in the thymus that the T lymphocytes are going to mature. Then exiting the thymus, we're going to have naive immunocompetent T lymphocytes. So T cells that are immunocompetent, that are able to do their job, but they're naive, meaning they have yet to be exposed to their specific pathogen. And exiting the thymus, we're going to have the two major varieties of T lymphocytes, the helper Ts and the cytotoxic or killer T cells. So let's talk about that T lymphocyte maturation process for a second. It occurs in the thymus, and it happens during just the early stages of life. So we're talking fetal period, infancy, young kid, okay? And a mature T lymphocyte is an immunocompetent T lymphocyte. And I was just saying that meant it's able to do its job. Well, what is its job? Its job is to respond to foreign antigens. And not any old random foreign antigens, but to respond to specific ones. And that's a really important characteristic. The other characteristic is the ability to tolerate self-antigens. So T lymphocytes that exit the thymus have to be able to do both these jobs. They got to respond to foreign things, then tolerate or not attack self-antigens. Self-antigens being the proteins that are marking one's own cells, one's own tissues. Now, surprisingly, most of the T lymphocytes that go into the thymus fail one or both of these tests. Only like 1% of the T lymphocytes end up graduating from the thymus. The other, 98, 99% are going to be killed in there because they're failing to do one of these things. Okay, so that is my T lymphocyte maturation. And when a T lymphocyte leaves the thymus, the T lymphocyte is going to be studded with receptors. So this is a helper T lymphocyte that is immunocompetent, that is mature. This is a cytotoxic T lymphocyte. And there's a variety of proteins on the surface here. We're interested in these T cell receptors that we've got. And a T cell receptor, there'll be maybe 100,000 copies of it on the T cell. But that T cell receptor doesn't respond to any old random foreign thing, but to a specific foreign antigen. So a immunocompetent T lymphocyte is going to have a specific type of T cell receptor that lets them respond to a specific foreign antigen. And that forms the basis of the specificity, which forms the basis of the memory and the improvement that's fundamental to the adaptive immune system. Okay, what about B lymphocyte maturation? Well, it occurs in the red bone marrow. So T in T lymphocyte stands for thymus. It's helpful to think that the B in B lymphocyte stands for bone marrow. It doesn't actually, but it's fun to pretend that. Now, B lymphocytes that exit the red bone marrow, of course, they're going to be mature, they're going to be immunocompetent. They're going to do the same two things. They respond to foreign antigens, and they tolerate self-antigens. Okay, what about the next step for these immunocompetent lymphocytes? You probably have about 1 million different varieties that are made early on in life. And those immunocompetent lymphocytes, whether they're B's exiting the bone marrow or T's exiting the thymus, those guys are going to migrate to areas where they're going to meet pathogens, such as 3 organs I have pictured here, which I'm sure you could recognize. Got some what's? What are all these colorful things? These are all lymph nodes. So of course, lymph nodes are a site of pathogen contact, of antigen challenge, and so lymphocytes will go there. What about these guys? What are these things? These are tonsils. And what tonsils are they? Palatine tonsils. So lymphocytes will travel to the tonsils, including these palatines. But of course also the lingual tonsils and the pharyngeal tonsils. Plus, lymphocytes will go to this guy just to the left of the stomach. What is this guy? He, of course, is the spleen. So these are major spots where we're going to find lymphocytes. And if we have 1 million different varieties of lymphocytes, that means they've got 1 million different kinds of receptors, with each lymphocyte variety having its own specific T cell or B cell receptor, letting us respond to 1 million different foreign antigens. Kind of crazy when you think about it. Okay, what about antigen-presenting cells? Well, in order for lymphocytes to do their job, lymphocytes have got to be presented with antigens, especially the T's. This is so important for the T's. T's lymphocytes need to be presented with antigens. And we have 3 cell varieties that do this job of antigen presenting. And there's cells we've met all 3 of these before. We've met macrophages, of course. We've learned about the fact that they're monocyte derivatives, that they're in the spleen and the liver and all sorts of places. They're free and fixed varieties. In AP1, we learned about phagocytic cells that are found in the skin, among other places. These are called dendritic cells. So macrophages are antigen-presenting cells. Dendritics are antigen-presenting cells. And lastly, B lymphocytes are antigen-presenting cells. So what all three of these guys will do, they will all engulf an antigen. So here's an antigen. It is being phagocytosed. It is then being chopped up by the digestive enzymes in a lysosome. And then pieces of that antigen are displayed on the surface of the antigen-presenting cell. So look at those little red triangles. Those are pieces of the foreign antigen. And they're being displayed by these proteins. Only antigen-presenting cells are going to have these proteins. They're called MHC2 proteins. And these purple blocks here are the MHC2 proteins. And only antigen-presenting cells have them. So antigen-presenting cells swallow and destroy foreign antigens. They destroy them by chopping them up. Then they hold them up on their surface. It's like a way of saying, hey, I just killed this thing. Check it out. Now, bear in mind, there is a fundamental difference between my first two antigen-presenting cells, which are my macrophages and my dendritic cells, and my final one, which is my B lymphocytes. The fundamental difference is what type of antigen they are going to engulf. Macrophages and dendritic cells, all they care about is that the antigen is foreign. But the B lymphocytes, when they're playing their role as antigen-presenting cells, they are engulfing specific antigens for which they have the receptor. All right, that's a crucial point. Keep that in your brain. So here we see a macrophage, and of course a macrophage is an antigen-presenting cell. Here is an antigen right here. It is foreign. That's all the macrophage cares about. Now notice the macrophage, he's an antigen-presenting cell. So on his surface, he's got these class 2, these type 2 MHC proteins. The macrophage is sticking out its pseudopods to begin with the engulfment, the phagocytosis. There's the engulfed antigen, the phagosome. The phagosome is going to fuse with a lysosome, which contains digestive enzymes, chops up the antigen. We see the bits and pieces here. Then those bits and pieces are complexed with an MHC2 protein, and the MHC2 protein is then stuck into the membrane on the surface. Now, to whom are we presenting the antigens? We said a macrophage was an antigen-presenting cell, and we see here to whom the presenting is happening. I don't know if that's good English or not. But it's a helper T cell. A helper T cell is the one that is presented with the antigen. And of course, it's not any old random helper T cell. It's the one with the correct receptor for that particular antigen. All right, let's walk through this in even greater detail. But before we do that, all right, as we're going to discuss adaptive immunity, we're going to be aware that there are two basic branches of it. There's antibody-mediated immunity, which is also called humoral immunity. It deals with extracellular pathogens. These are pathogens found in blood, in lymph, in interstitial fluid. B lymphocytes and plasma cells are the major cells involved in antibody-mediated immunity. By the way, the word humoral means having to do with humors, and humors is an old-fashioned word for body fluids. And of course, body fluids are where we would find things that are extracellular. Now, it stands to reason that if we have one branch to deal with extracellular pathogens, we'll have another branch to deal with intracellular pathogens. So bacteria or viruses that have invaded our body cells. And this branch is called cell-mediated immunity. And the primary actor in this branch are the killer T cells, aka the cytotoxic T cells. Now, another important fact is that helper T cells are going to control both of these branches. So they're the ones in charge. Okay, so let's figure out how we activate helper T cells. All right, this guy, notice he is labeled APC. That stands for antigen presenting cell. So let's say he's a macrophage, okay? This macrophage has engulfed an antigen, chopped it up, and now it is presenting that antigen on its surface via these MHC2 proteins. Now this macrophage is going to wander on over to a lymph node, perhaps, or someplace with a population of helper T cells. This is a naive helper T cell. He's naive, meaning he has yet to respond to an antigen. So this is going to be an initial response here. We can tell he's immunocompetent because we can see he's got his T cell receptors. And of course, he doesn't have a multitude of different types of receptors, but just many copies of a single receptor, which allow him to be specific to that receptor. And this specific helper T cell binds to that specific antigen fragment being presented by this macrophage, which is an antigen-presenting cell, via its class II MHC protein. Now, in response to this presentation, the helper T cell is going to start releasing some chemicals. Now, the chemicals released by helper T cells are known in general as cytokines. More on them in a second. What we also notice this helper T cell doing is he is dividing. He is doing mitosis. It's like the macrophage says, hey, I just killed this thing. Does anybody recognize it? Oh, you do. Great. And then this helper T cell is like, all right, let me form an army to direct the response against it. So here's just the beautiful point here, okay? In doing its division, the helper T cell actually is going to make two varieties of cells. It makes activated helper T cells. Activated helper T cells are for right now, okay? To help deal with and direct the response against this particular antigen right now. So let's say these two guys are activated helper T cells, all right? But this guy over here, this guy is not purposed for the current response. This guy is a memory helper T cell. He is there for subsequent exposures, and his production underlies why our response to subsequent exposures is so much faster and stronger than our initial response. Okay, so here is our activated helper T cell. Remember, we presented the antigen to a naive helper T cell. The helper T cell, the naive one, then divided, making activated helper T cells for right now and memory helper T cells for later. This is an activated helper T cell for right now. he is going to direct the immune response of that, or rather to that antigen. Now, he does that primarily by releasing chemicals. And those chemicals, as we just said, are known as cytokines. Cyto, of course, means cell. Kines, think about like the word kinetic, like in kinetic energy. So cytokine means cell movers. And cytokines, what they're going to do is they are going to promote the response of other adaptive and innate cells to help deal with this particular bacterium that we are currently infected with. Notice that this cytokine, it says, for example, IL-2. IL is an abbreviation for interleukin. Interleukin, let's think about that word. Interleukin, inter means between. Leukin refers to leukocytes or white blood cells. So these are messages between white blood cells. So the helper T cell is going to activate other white blood cells, which of course are the soldiers of our immune system. And there are a whole bunch of different interleukins that with all sorts of different jobs and different things they do that we're not even going to pretend to get into. Okay, so we've got helper T cells ready to direct the response. Let's throw somebody else into the mix. Let's suppose a B lymphocyte, a naive B lymphocyte, grabbed onto that same antigen. Remember that antigen the macrophage had? A naive B lymphocyte grabbed onto and swallowed that same antigen, chopped it up, and is displaying it on its surface. Here we see it displaying it on its surface here, okay? And on its MHC2 proteins, of course. And by the way, it wasn't random that this B lymphocyte swallowed that antigen. This B lymphocyte has its own receptors for it, specific receptors. Now, once the B lymphocyte engulfs and presents that same antigen, the B lymphocyte then goes looking for one of those activated helper T cells, of which we just made a whole bunch. The B lymphocyte is basically saying, hey, I recognize this guy. I was specific to him, so I killed him. Do I have permission to then go and kill a whole bunch more? So the B lymphocyte, who specifically engulfed this antigen, is then showing it to that activated helper T cell that is also specific for the same thing. The activated helper T cell is responding by releasing an interleukin, giving this B cell permission to respond. So it's like a double check. It's like a safety check to prevent the B lymphocytes from, running wild and killing things they should not. Now, once the B lymphocyte has gotten permission, the B lymphocyte is then going to divide and check out what this division results in. The division results into activated B cells for right now. These guys, the activated ones for right now, are called plasma cells, and they work by making antibodies, which are specific immune proteins. But then the B lymphocyte that got permission and started dividing is also making memory B cells for next time and the time after that. So our response will be stronger and better upon subsequent exposure. Okay, the antibodies are going to do the work against this pathogen. And by the way, remember, antibodies were part of what branch of immunity? Antibody-mediated immunity or humoral immunity. So these antibodies are going to help us deal with that original pathogen if it's in any sort of extracellular fluid, blood, lymph, interstitial fluid, etc. Now, how do antibodies do that? Well, let's take a look. An antibody is a Y-shaped specific immune protein released by plasma cells. Antibodies are also called immunoglobulins, or just Igs. They're also known as gamma globulins. Now, antibodies have two spots upon which they can bind to antigens. They got two antigen binding sites here and here, kind of like imagine a person with their arms out like this. I didn't want to draw an arrow there. Let's get rid of that arrow. Imagine a person here with his arms out. Boom, 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 boom. And the two hands are where it's grabbing onto antigens. And not any random antigen, but a specific one. Now, the binding of that antibody to its specific antigen can do a bunch of things. One thing it can do is just cover up the dangerous part of a microbe or a toxin. And if in that case, the antibody is doing some neutralization, it's neutralizing the microbe or the toxin, it's preventing it from doing whatever harm it could do. Antibodies also, by grabbing onto more than one antigen, it can link them together, forming a clump. And we've seen this in talking about blood typing and blood donations gone wrong. That's called agglutination. The idea being, if we clump the bacteria, number one, it's going to slow them down. Number two, a clump is much more likely to run into a macrophage and get engulfed. Now, if we are using the term agglutination, notice that is only utilized for the clumping of cells. If we are clumping non-cellular things, like particles, maybe toxins released by a bacterium, then that type of clumping is called precipitation. And that also is going to facilitate phagocytosis and restrict the movement of the toxins. Okay, other things antibodies can do. When antibodies bind onto bacterium, they are going to expose part of themselves, like the feet of the antibody here is being exposed. Again, if the hand, remember the hand, it's like a person, and the hands were doing the grabbing, and now the feet are exposed, and the feet can activate the complement protein system, which we talked about last lecture. So go back and review that. Also, sometimes when an antibody grabs onto a bacterium, it changes its structure and it exposes a portion of the antibody that is really attracted to, or rather phagocytes are really attracted to it. So if a phagocyte bumps into that part of an antibody, when the antibody has grabbed onto something, that makes it almost a certainty that the phagocyte is going to do some phagocytosis. And remember, that was called opsonization. And then finally, antibodies are going to activate natural killer cells, which are great at killing cells, our own body cells that have become infected. So these are all the wonderful things that antibodies, these immune proteins, are capable of doing. And let's look at a graph. I know you guys love graphs, so it's good we got one here. And I just want to look at the red graph here, okay? So the red graph, and we've got two graphs here, right? We're only looking at the red trace on both these graphs. Notice the first graph, it says primary response. Second graph, it says secondary response. And looking at this red line, it says IgG. IgG are a specific type of antibody. So we're looking at levels of this type of antibody. That's what's on our y-axis here. And then days from exposure is on our x-axis. And exposure happened at day 0. Then it's not till like a week before the IgGs start getting made, it'll last for another variety of antibody. This is called the lag phase. So everything we've just talked about, like the whole part about the macrophage swallowing the antigen, chopping it up, finding the right helper T cell, letting that helper T cell divide, then the B lymphocyte binding that same antigen, finding the right helper T cell, then the B lymphocyte dividing, then the plasma cells making antibodies. All that happened in that lag phase. Now, how do we feel during this lag phase and a little bit beyond it? Well, we're going to have symptoms. We're going to have symptoms, absolutely. And look at this red line here. It's going up, 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 up. Took about 2 weeks to reach its peak, and then it goes down, 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 down. Pretty like symmetrical up, down here. That is our primary exposure when we've got naive helper T lymphocytes and naive B lymphocytes. Now let's compare that to our secondary response. First of all, look how much shorter the lag phase is before antibody levels start to go up. The lag phase is far reduced because we've got memory cells. And memory cells do everything better, faster, and stronger. And then by like day 4, our antibody levels are so high. And then they stay high. They don't go down kind of quickly like they did in the primary response. They stay high for a while. So the antibody levels, like, look how much bigger it is. So it's like how big here? Like that. I need a ruler to measure this, don't I? And then it's like that. What is that, like 3 times bigger? Well, it's actually even more because this y-axis is on a logarithmic scale. And basically what that means is if this secondary response is 3 times higher, we actually have to take it to 10 to the third. Not just 3 times, but 10 to the third times higher. And 10 to the third times higher is 1,000 times higher. So our secondary response is so much more robust than the primary. Way higher antibody levels, way shorter lag phase. And by the way, the antibodies are better. They grab on better. They do all their jobs better as well. And this is the fundamental basis of immunity. This is how immunity works. We make memory cells. Memory cells let us do everything better on subsequent exposures. So that's really what immunity boils down to. Okay. Let's actually classify some different types of immunity. So there's 2 ways we can classify immunity. It can be natural or it can be artificial. It can be active or it can be passive. And I want to run through 4 scenarios with you and see if you can figure out what word, natural versus artificial or active versus passive. See if you can figure out what word is going to apply for both these categories. So in this first one here, let's suppose somebody was just, in the course of their life, they got infected with this bacterium. This is some connective tissue from under their skin, and this is the bacterium which is very present there. So let's suppose the person just got infected, normal course of their everyday lives, and in response to getting infected, they made their own antibodies. They made their own memory cells. What kind of immunity do you think that is? Do you think it's natural or artificial? Well, if it just happened because they were just living their lives, this is an example of natural immunity. And then if that person, in response to the infection, they made antibodies, they made their own memory cells. That sounds very active to me. So if you get an infection and you make memory cells and antibodies in response to it, that is an example of natural, I should probably spell natural correctly there, natural active immunity. Okay, that was scenario number 1. Let's do scenario number 2. In scenario number 2, let's suppose somebody is cleaning their garage and they see this cute little spider and they pick it up and they're playing with it because it's really pretty. And then it bites them. And this, of course, is a black widow spider. Her venom has some very toxic proteins in it. Now, we don't have time for the body's response. We can't wait for the body to respond. The body is going to respond like it did up here, but we don't have time for it. So luckily, somebody takes this person to the local spider bite clinic, and they get an injection of antibodies. So antibodies are injection of antibodies. I'm just going to write IGs because it's easier to write IGs. So with an injection of antibodies, does that sound natural or artificial? That is, in fact, artificial. Now, does it sound active or passive? Like the person who got bit, they didn't make the antibodies that were injected. So this injection of antibodies, like for example, in response to a spider bite or a snake bite, that is artificial passive immunity. Okay, so we got 2 scenarios giving us 2 varieties. We had natural active and then we had artificial passive. Let's do 2 more scenarios. 2 more scenarios. All right, here we have a fetus. Fetus is in mom's uterus. There's the placenta where things are exchanged between mom and fetus. And one of the things that happens is antibodies, so antibodies, immunoglobulins, IGs go from mom. IGs go from mom to fetus. And that happens at the placenta. And these antibodies are going to protect that fetus because his immune system is still developing. Now, does this sound natural or artificial? This, of course, is totally natural. Now, is the fetus making its own antibodies or just receiving them? He's just receiving them, so this is going to be passive. Now, this is not the only way natural passive immunity can occur. Can you think of the other one? Well, antibodies will also be secreted into mom's breast milk. So that is another route for antibody delivery to the baby, another way for natural passive immunity to work. All right, our final one. This is a killed virus. If there was an injection of a killed virus or a weakened virus or a harmless look-alike virus, or substitute the word bacterium in there as well, the injection of a killed, weak, or look-alike virus. In response to that, the body makes its own antibodies and its own memory B cells. This gives us immunity, but spares us the symptoms. This, of course, is the fundamental idea behind vaccination, which is one of the greatest inventions in mankind. Don't believe anybody who tells you different. They don't know what they're talking about. And does this sound natural or artificial? Well, it's an injection, so it's artificial. But then the body is making its own memory cells and antibodies, so it is artificial and active. And those are the 4 ways that B cell immunity is created. Okay, that finishes up the antibody mediated branch, the humoral branch. Let's now deal with the intracellular one, the cell-mediated immunity. This is how we kill pathogens that have invaded cells. Also, it's how we kill cancerous cells. And the primary actor in cell-mediated immunity are the killer T cells. Okay, looks like there's a lot going on in this picture. But what we got to do is realize that almost every cell in your body, every healthy cell in your body, the only exception, by the way, are red blood cells. But everybody else, every healthy cell is always going to make something called MHC1 proteins. MHC1 proteins, what they do is they display pieces of self-antigens on the surface. So a normal healthy cell, we don't want to kill it, right? A normal healthy cell is uninfected, is non-cancerous. It's going to have normal healthy self-antigens. So here are a pair of normal healthy self-antigens being displayed on this MHC1 protein. So everybody is going to be doing this. Anytime a normal healthy cell makes proteins, it chops up a few, puts them on their surface. This is a way of saying, hey, I'm healthy. It's a way of telling the immune system to leave me alone. Now, red blood cells don't do this because, of course, remember, what are red blood cells missing? A nucleus and organelles. But everybody else does that. Everybody else, every other healthy cell in your body is always displaying fragments of proteins, fragments of self-antigens on its surface as a way of creating signage that says, hey, I'm good, I'm uninfected, I'm non-cancerous, leave me alone. All right, so that's what's going on in this diagram here. I just want you to have that fundamental idea that healthy cells are displaying pieces of healthy proteins as a way to say, I'm healthy. And they're doing that display on MHC1s. Remember, we dealt with MHC2s earlier. Okay, but what happens if a cell is infected with a virus or it becomes cancerous? So here is a cell that was infected with a virus. We know viruses try to take over, they try to hijack the cell's machinery to make more viruses. Well, if the cell has become infected, it's going to start making viral proteins. And now it's displaying viral proteins on its surface. Displaying viral proteins via its MHC1 molecules. This is a way to say, hey, I've been infected with this particular virus. Now, who are we displaying these for? They're displaying it for a naive cytotoxic or killer T cell. So here is the infected cell. The infected cell has got viral antigens on its MHC1s. It has bumped into a naive cytotoxic T cell. Not any old random one, of course, but one with the correct receptor, the specific receptor. In response to this binding, this naive cytotoxic T cell is going to divide, making activated ones for this infection plus memory ones for next time. By the way, notice that we got like some chemicals swooping in here. some interleukins. Those would have been released by helper T cells that are directing the response to the same antigen. So the T cell is not only being stimulated by the viral fragment on the infected cell, this cytotoxic T cell is also getting permission from the director, which is the helper T cell. And in the cytotoxic T cell, the killer T cell divides to make active ones for right now and memory ones for next time. Now the active killer T cells, they go find more infected cells, more cells displaying that same viral protein on their MHC1s, and the killer T cells kill them, either by releasing some proteins that punch holes into the cell, causing water to rush in and letting them explode, or by causing the infected cell to commit apoptosis or cellular suicide. And so this is how we kill cells. Viral infected cells or cancerous cells. If this cell had become cancerous, it would not be displaying normal proteins, and we'd be killing it. So that is the basic way killer T cells kill infected cells. It's also how they know how cells have been infected. And remember, we are creating memory cells too. So our response subsequently is more robust, better, faster, stronger. Any adjective you want to use like that. Well, not any adjective, not slower, weaker. Anyone that means better, stronger, faster. Okay. The final thing we're going to talk about is one other branch of T cell. There's also T cells called regulatory T cells. They used to be called suppressor T cells. I kind of like that name better. Regulatory T cells, they release cytokines, interleukins, and other things that suppress immune activity. They're going to do this, ideally, once we've defeated whoever is currently infecting us. Because we don't want immune cells going wild, because then they can start attacking our own body cells. And regulatory T cells help us prevent that from happening. And with that, we are done with the immune system. Lecture number 2 might have taken me a little longer than I had anticipated on it taking, but that's okay. I had a good time. Anyway, let me know if you have any questions, as always, and I'll see you next time. Bye-bye.